This is a production of Cornell University.
Well, thank you very much for the invitation to speak today. I recognize quite a few faces here. Great to have you here. Welcome to nearly the end of the semester and our first snows. So I'm going to be talking with you today about digging deeper, literally and figuratively, into soil health. And I thought a lot about what I wanted to present for today, what would kind of work, and I kind of landed upon kind of following a bit of a story of my journey and these lessons learned from working in diverse ecosystems, when a lot of our soil health framework really comes from agricultural soils. So just all these lessons I've kind of picked up over time, which brings us right into kind of my non-linear journey. Oh, it's not, there we go. My non-linear journey of getting here. So been very much all around in my journey. So this has taken me, not only do I work in diverse ecosystems, but I've kind of worked in ecosystems all across the country and also in Canada. And so this has really broadened my experience, and hopefully the lessons that I've learned through this I can convey, and hopefully some of them at least are helpful to folks in this room. So just a little bit of kind of walking through. I'm not going to cover everything. I'm going to cover the things where I've kind of learned the most lessons over my span of research. So I started at the University of Washington, got my master's working in forest soils. So this was, I really, along with that non-linear road to get here, I really didn't know what I wanted to do. I was in environment, I was in philosophy, I was in all kinds of different things. And kind of happenstanced onto soil, but just I wanted to do a lot of different things. I wanted to do biology and geology and chemistry and all of these different things. And I realized, hey, soil covers all of that. And it's so complex, it's living. I'm probably not going to get bored. And it's worked out so far. There's still a lot to learn, which you'll see. I have lessons I've learned, but a lot of questions still remaining. For my PhD, I was at the University of Alberta in Canada. I wanted to kind of dip my toes into agriculture, so I chose an agroforestry project. Still had that tree component, which I liked that complexity, but wanted to kind of get into agriculture a little bit, working with biochar. So spent 5 years there. This is me being extremely happy that the trees we planted had surpassed my height, although it's not that much of a feat. But I was still happy after a couple of years that, it had surpassed my height. So now I've been at SUNY ESF as an assistant professor for a couple of years now. I'm working across various ecosystems. So I work in short rotation woody crops, forest soils, and getting into silvopastures. So we'll talk about those. And then we'll talk silvopasture, which is a very large new multi-farm initiative that I'm working on with a lot of collaborators from Cornell. So we'll kind of get into that and how these lessons over time in soil sampling, soil methods, understanding soil health will kind of feed into how we're planning the silvopasture project and then just in the future. So let's talk soil health. So soil health has really been evolving over the last, especially decade, and really growing quickly with a lot of public support behind it. And part of that is that we have these really cool ways of demonstrating it. It's more intuitive for people to understand that these are living systems, soil health like human health, and it's very demonstrable. We can, so these were buried underwear pants with elastic bands. And so you could really kind of see over time how if you had a healthier soil with happier microbes, more organic matter, then you would have, what's this laser pointer? Yeah, then you would have more of your decomposition of that cotton material. So, soil health has just really been expanding, but a lot of it is still, based on agriculture. And so we're missing some of the things that are in other ecosystems, which then would apply to understanding how management in these other diverse ecosystems affects soil health. So when we talk about soil health, I know a lot of you in this room probably know this, but we're talking about the biological, the physical and the chemical, how those all work together. In the past, we've really focused a lot on the chemical side. And so now we're really focusing more on the biological side and understanding that this is a whole system working together. And so your biology affects your physical characteristics, which affects your hydrology, which affects your chemistry. So all of it is working in tandem. And then we've developed several, I left those on for my students. Well, you guys can help me. Who wants to shout out the soil health principles? Maximize living roots.
Minimize disturbance.
Minimize disturbance.
Maximize soil cover.
Soil cover, maximize soil cover, and maximize pollinators, biodiversity. Yep. All right. Awesome. Thank you all. So, we have our kind of general soil health principles, right? And so part of the reason that this has been in agriculture as well is that we kind of see other systems as largely meeting these requirements, but we still have, or these foundations, but we still have, room where we can improve and understand how management affects other systems. So our principles in terms of what we can be doing, again, largely based in agriculture. One of the things that interested me about working in silvopasture is just that it covers most of this. So silvopasture really covers a lot of our soil health principles. And again, similar to why I started in soil, it's just like one of the most complex systems that you can work in. And so that is interesting to me. So my talk here and my work over time has largely focused on soil organic matter. So it will be soil organic matter and the carbon it contains, heavy in terms of my research and what I'll be talking about. But soil organic matter is really synonymous with soil health. So soil organic matter is really the figurative and literal glue that holds soil together. And basically everything that we look at for soil health is affected by soil organic matter. So biodiversity and function, water storage, filtration, nutrient storage and production. So this will be very soil organic matter heavy in terms of this seminar and the lessons I've learned, but it really applies to other things as well, like soil sampling and methods in general. Sorry. Oh, another thing about soil organic matter is that it's also easy to demonstrate, and you can kind of see, you can show people. So I like that, again, when we're speaking to farmers and producers, we can really show them, they can see on their own farm if they have good soil health and a lot of soil organic matter, if they're building that over time. So these are, 3 of these are from an agroforestry system. And so does anyone call out the one that's not from an agroforestry system? Maybe a forest? Which one's different up there? Which one doesn't belong?
Second.
The second one, right? And so the other 3 are from a woodland, grassland, under woodland, under grassland, and under annual cropped land in an agroforestry system. What do you think the first one is? Annually cropped and tilled under grassland or under woodland, same soil.
That's grass.
What's that?
I think it's grassland.
This one is actually, this is the annual cropped and tilled. This is your grassland. So that deeper carbon profile that you can see there, more organic matter going deeper. And then this would be under your woodland, and you've got that alluviation. So those are all the same soil, pretty close to each other. And so that, like, I love to be able to demonstrate that to people, and I think that's a real strong benefit. Can also demonstrate it in other ways. So these are just some nice little experiments that we've done with kids for soil organic matter, and just showing how it holds nutrients like ketchup, if there's nutrients in ketchup, and helps with soil structure, and especially when that soil gets wet. So very cool ways to demonstrate this. And this is a lot of the reasons why the soil health framework has really help take off and help engage the public a lot, which is what we're trying to do with our larger projects that we're developing, is engage the public around these and producers around these principles. All right, so let's jump into soil carbon storage and cycling and what can essentially change that, because that's a lot of what this will focus on. So essentially we know that soils contain a lot of carbon, more than the atmosphere and vegetation combined. And historically, they've been acting as a carbon sink for us. So this is changing in different ecosystems and in different places around the world where we might have soil switching from a carbon sink to a carbon source. And there are various reasons for this. So land use change, land management, climate change are all affecting whether soil in certain regions, in certain ecosystems are still acting as carbon sinks for us or acting as sources. So this gets us to our first case study from my master's in the Pacific Northwest, so looking at forest management. So I worked in a Douglas fir plantation, and generally speaking, when we look at secondary forests and plantations, you know, we initially, when we have a harvest, we initially have some soil organic carbon loss, and we obviously have that loss of above ground biomass carbon as well. How greenhouse gas emissions are affected really depends on management and other things that are going on, but this is kind of our general overview. Usually we can recover pretty quickly from a harvest, but again, it's dependent on the soils and the location, how it's done. So as I said, I focused in a Douglas fir plantation, and we specifically looked at thinning and with or without nitrogen addition. And we sampled very deep soil, so very deep soils in the Pacific Northwest. It's one of the reasons so productive there. This is a 40-year-old Douglas fir plantation, and it was very difficult to dig. I dug, I think, over 30 pits down to 1.5 meters. So when a storm came, I basically jumped into my own grave that I had dug to protect myself, and it worked. Luckily, I did not end up in digging my own grave, but lots of rocks, right? So forest soils, we tended to, if there were a lot of rocks, slopes, those are our forest soils, right? And if it was good, flatter land, those are our agricultural soils. So there was already some selection that went into this. So forest soils are notoriously hard to dig. They also have a lot of spatial variability. So how much do we really need a sample to capture that and capture change over time in soil health and soil carbon? And one of the things as I was out there digging these holes is that I was using a hammer core. So here's what a hammer core looks like at the top. And that broke on me because I think it was my first day out there. It was the only method I knew at the time. Immediately broke. I'm out in the middle of nowhere because it hit a rock. I still have them, a bunch of those parts in my office because I went through so many of them out there, but immediately had to come up and adapt and come up with a different method. So started using the clod method to get bulk density. So this is to get bulk density to understand how much mass of soil is in a given volume, including those pore spaces. So it's very important to be able to know how much carbon we have overall. So there's other methods too, like augers, probes, and then you have full pit quantitative excavation. That's considered kind of the golden standard for getting bulk density. And then you have a regular hole. So when I was out there and I had to immediately switch what I was doing because my tool broke, I was told that those methods are interchangeable. So it's fine, just, go ahead and use the core when you can, use these other methods. And I thought, well, are they really interchangeable though? And I couldn't find many studies on it. So adapted this and did this study in addition. So this is a lessons learned. Are these interchangeable? So no, I did the 3 different methods, like a excavation or a volumetric clod and hammer core, and they were not interchangeable. So the hammer core seemed to consistently underestimate bulk density. So bulk density, especially working in New York soils, is just a real pain, but it's incredibly important especially for anything carbon related, but anything where you're trying to just know the total mass of nutrients or total mass of something. It also, in and of itself, can be an important soil health metric. How compacted is your soil? So this is a problem because the hammer core is one of the most common, easiest to use, but you can't really use it in these diverse systems. It also has problems within agriculture. When we're using these methods, we can still have biases with collecting bulk density the way that we do it. It takes a lot of time. And there are other problems such as biases when we have tilled or no tilled. So this kind of took me on a rabbit hole of looking into all of these problems and kind of discovering things that didn't seem to be very well advertised, I guess you would say, at that time. So essentially in agriculture, when you till or do not till, you can compact or fluff up your soil. So these are the bulk densities I have highlighted there. So 1.2 versus 1 now, so it's fluffier over here. And then if you sample to 30 centimeters at time 0 and 30 centimeters at time 1 after you've tilled your soil, you're actually sampling less mass. And so this can lead to a bias and an underestimation of how much soil that you sampled, and then that changes your final calculations. So bulk density and the way that we calculate it are all highly challenging, especially in New York soils. There are some ways to overcome this. So I use the equivalent soil mass approach. It's been much more widely adopted at this point over the last couple of years to overcome these biases and to be able to calculate our carbon properly. It helps a little bit too with the biases and how we're collecting our bulk density and all the issues around that. Doesn't help a huge amount when we're hitting all these rocks and other things and just not collecting all of our soil. But this is a helpful method that I've adopted fully over time, but still have the issues with bulk density that we'll kind of get into later with other iterations of projects. But if you talk to any soil scientists who are out there in the field, particularly in the Northeast, bulk density is just a huge pain. Or if you've been in the field doing it, you know that it's a huge pain, but it's critical. So back to what we found, just looking at these soils and forest management. So we found that even in the deeper soil, thinning had reduced our total soil carbon stocks were lower in our thinning treatment versus our control. So this really got me thinking about deeper soil and how even over this 40-year time frame, we can affect deeper soil. I should caveat that we did not have original samples from 40 years before this to make sure that these soils actually started on the same playing field. So, it's observational. It's always better to have your baseline. But that was another reason I took a deeper dive into deep soils, because I wanted to know how often are we seeing effects in deeper soil. And in agricultural soils, we often are only sampling the surface, especially when we're looking at soil health. So these deeper soils soil tends to be sampled deeper, more in forest soils, or especially when we're looking at soil genesis. So what does the subsoil have to do with it? Well, it contains most of our carbon. And contrary to kind of adopted opinions for a while, it's active. So especially when you have roots that are deeper rooting plants, woody plants, you have roots all the way down to a meter, all the way down to multiple meters, and you have microbial biomass, you have life, you have activity that's also going deeper into the soil and processing that organic matter. So you have activity, you have turnover of organic matter even in deeper soils. But over time, we have been sampling more and more shallow. 25 centimeters was the average in 2019 for most studies sampling soil. So we really have this lack of understanding of what's going on in deeper soil. And this was even in forest soils as well, not just agricultural soils. So this took me into a deeper dive of really understanding how carbon is being cycled in soils, what's going on. And this has been evolving over time of our understanding in soil science of soil organic matter. A lot of work being done here for soil organic matter, how it's cycled, and that essentially what we have is over time we have this downward cycling of soil organic carbon. It's being processed by microbes, and we have this dissolved organic carbon pool, and it's largely from microbial processed inputs from the rhizosphere. And that's all being cycled over time. So as we go deeper in the soil, we have more microbial derived compounds, more mineral associated organic carbon, and the age of our dissolved organic carbon and soil organic carbon gets older. So it's not intrinsically stable, this deeper carbon. It's just been cycling a lot longer, and it's at different stages. So we had this old view of it being stable, especially this deeper carbon, and that we had this pool of carbon that was stable. But now we really understand that it's not necessarily stable. If you have microbes that can process it and the conditions allow for that, they are going to process it and decompose it. So deep soils is something I'm pretty passionate about sampling. But in the Northeast, we actually don't have as deep soils as I'm used to in other places that I've been. So don't have to go quite as deep here because you typically hit some sort of impenetrable layer, which is kind of nice to some degree. But you're hitting a lot of rocks before you get there. But that's something that in agricultural soils, we don't have the effects as often below just the surface soil. So it's just missed a lot, and we have this gap in our knowledge. And especially in something like a silvopasture system where you're planting trees, anything where you're going to have an impact on deeper soils, we need to be sampling deeper soil. And we don't really have any sort of soil health framework for deeper soil. Most of the tests are just being done on the upper 15 centimeters of soil. So we have this complete lack of knowledge about what our soil health framework for deeper soil. And roots are tapping into that soil, so it can certainly affect our productivity. The structure of that soil as well can affect our productivity. So now looking at cultivation, so in cropland, we have loss of carbon, we have increase in greenhouse gas emissions. So looking at management, so this is another thing I looked at, looking at different types of management and specifically looking at biochar, which can increase carbon and reduce greenhouse gas emissions. So this was my dipping my toes into agriculture. I'm not going to get too much into this just because it is directly in agriculture. We have a lot more information on this. I didn't learn massive lessons from this in terms of my overall how I'm performing soil sampling and other things, but it was interesting. Biochar did only affect the surface soil, but it stayed there, whereas our manure compost that we put in, which is from what the biochar was produced, completely was lost after a couple of years. So kind of did what we would expect. A lot of the research out there shows that biochar is more persistent in your soil and can reduce greenhouse gas emissions. So long-lived biochar, it did what we thought. And it really did only affect the surface soil. So we applied it to the surface soil. It did only affect the surface soil. So to some degree, it's not that we always have to sample deeper. We really have to understand what we're aiming for, what we think we could be affecting and why with what we're planting or what treatments we're using. So just to caveat that with, it was probably 40%, maybe even 50% more work to, sample those deeper layers that really didn't have any changes. So we could have done more sampling or a broader system or broader experiments without sampling as deep. So in this case, I wouldn't say it was a waste of time, but maybe our time could have been spent better elsewhere rather than sampling as deep in this particular system. Let's see, so let's get to agroforestry. So agroforestry, we know can increase carbon below ground, increases carbon certainly above ground, and can reduce or does reduce greenhouse gas emissions. So agroforestry, combining trees and shrubs into cropland, intentionally putting trees and shrubs into cropland or leaving trees and shrubs in cropland. Can also integrate animals. All right, so we worked in hedgerow and shelter belt systems, which are really linear features, woodland, cropland, grassland. I'm gonna try to go fairly quick here so I can keep going. Couple of the interests, so we did augering instead of a hammer core. This seemed to work really well in these non-rocky soils for getting accurate bulk density. Used the equivalent soil mass approach, looked at heterotrophic, autotrophic respiration, and measured deadwood, which really hasn't been done in agroforestry systems before. So what we found is that agroforestry, again, was really only affecting the surface soil in terms of soil organic carbon. We had an increase in our woodland, but overall was not affecting our soil organic carbon across the whole profile. Obviously, a lot of carbon above ground. But what was really interesting here was within the woodland, the difference between the hedgerow, which is this legacy woodland with deadwood, and the planted shelter belt. So you can kind of just see by the pictures how different those look. The hedgerow has been left and has this deadwood. And so this deadwood component of the hedgerow actually was highly correlated with carbon cycling in general. So what we found is that basically all of the other metrics of, so total soil organic carbon, surface soil organic carbon, water soluble organic carbon, microbial biomass carbon, heterotrophic respiration, all related to the deadwood, all positively correlated with standing deadwood and coarse deadwood. So this really got me thinking about, you know, these mechanisms. What exactly is happening here? What are we looking at in the soil in terms of these mechanisms that are, if the deadwood is in fact causal for this, because again, this wasn't a direct causal relationship, just correlated. But if there was a cause and effect here, you know, what is potentially going on with the deadwood? And if we are increasing our soil organic carbon and our soil organic matter, is it going to persist? And what would be the conditions under which that would persist? So if we leave deadwood, are we really getting this persistence? Because we also had this increase in heterotrophic respiration. So this kind of gets us back to our new understanding of soil organic carbon cycling and the continuum model of that essentially, you know, as our inputs of organic matter are decomposed by microorganisms, it gets smaller and smaller. And has more potential for interactions and absorption. So we look at it as absorbing like organic associations, organic mineral associations, and then all of that can be formed into aggregates. And all of these things can protect our organic matter and allow it to persist more over time. So if we break that down just a little bit more simply, we're looking at particulate organic matter, dissolved organic matter, and then what we call mineral associated organic matter. Mineral associated organic matter is what we consider to be more protected organic matter. So that's kind of where things have gone, but then also this interaction between this protected mineral associated organic matter and aggregation. So aggregation can also physically protect, like a shield, protect that organic matter from microorganisms. And then we have this chemical protection from the mineral association. So combining those two, we can potentially have this really strong protection of organic matter. So I wanted to look at that more deeply, these mechanisms, and I thought, well, maybe there's this silver bullet, because if you kind of take all of those concepts and you think, well, clay is really what most of our organic matter is bonding with, is absorbed to. It provides a lot of protection in that sense, and clay can form these smaller silt-sized aggregates that don't even have pores so small that bacteria can't even get inside. So maybe this is a highly protected portion of organic matter, and we should create circumstances where we can get more of these more clay protected within silt-sized aggregates with organic matter associated with it. So I did a study directly looking at that across these land types, across the biochar and manure treatments that we did, planted it with blue grama grass, a C4 species, did a lot of fractionation. So this is a fairly novel technique to get to that very specific clay within silt size aggregates and the carbon associated with it. It was never ending fraction. So this is me during the pandemic, enjoying this many, many hours. I did get some cool artwork out of it though. So this was the soil on the sieves. The coarse silt actually, which had a lot of really what we would call particulate organic matter. So this special fraction I was looking for that I termed aggregated clay had a lot of carbon. So about 30% of the total carbon in the soil across the land uses and across the depths. So it's a pretty substantial portion of the carbon. So I thought, well, you know, if that stays there, if that's, you know, and maybe it stays more, it's more persistent in, you know, the woodland versus the cropland, right? Well, not so much. Lo and behold, priming. So this is when I planted this grass, essentially the exudates released from the grass and the microorganisms and their metabolism and all of that and their decomposition of the organic matter. What we look at for priming is with our control where we didn't plant the grass and our grass planted treatment, did the organic matter, was it decomposed more quickly when we planted the grass? And that kind of tells us if that organic matter is persisting or not. So essentially when we planted the grass, we had priming or an increased loss of organic matter from the exact fraction and only the exact fraction that would have been considered to be the most stable. So the aggregated clay is where we were really losing the majority of or all of that carbon. And that was consistent across the depths and across the land uses. It was also consistent for the manure and biochar just in the surface soil, except the biochar did seem to maybe protect it a little bit and maybe mitigate that decomposition within that fraction. So I said back to the drawing board, trying to figure this all out. One of my thinking on this is that essentially the microorganisms getting all this additional carbon from the roots, they create a lot of enzymes that then can get into those silt size aggregates and break them down. So then they release a lot of that carbon, break down those silt size aggregates, and then they're available for, that organic matter is available for decomposition. The biochar can absorb enzymes, and so maybe that was the mechanism for it working to help mitigate this. So this is still ongoing, and I think it's a really interesting line of research, but it really isn't practical. The amount of time and effort to fractionate the soil in this way was incredibly challenging. It would be very difficult to do on a large scale. So I'm trying to think of ways to do this in ongoing studies on a more like soil health framework where it can be done more in mass, and we can find the fraction that kind of represents this and is easier to isolate. And that's part of the problem with this fractionation. It can give us a lot of insight, but it is a challenge in terms of helping us to, in terms of us being able to include it in a framework where we can do it enough to get real answers across different ecosystems. All right, so current research, I'm gonna go through this pretty quick because it's kind of just getting started here in the last couple of summers. Working in shrub willow, basically another biochar project, but also we kind of a combination of the planting a C4 grass to trace new carbon inputs and be able to measure heterotrophic respiration. And then also looking at if the biochar can mitigate losses of organic matter after we've taken out this shrub willow and maybe are transitioning that into a grassland or a cropland. So removing a woody plant that roots deeply and creating a cropland or a grassland was kind of the idea of this, because a lot of folks, after they do a short rotation woody biomass for one life cycle and then maybe transition back. Sometimes this is just to improve soil health. So we wanted to see if we could mitigate losses after you do that. So this is a lot of trial and error, a lot of rocks in this. We had like gophers too, creating holes under the ground. So it was very difficult to measure bulk density. We also were trialing a new Lycor equipment to actually measure stable isotopes, which was a lot of trial and error as well. But one cool thing that came from what we have so far is looking at, so we did the Slakes app to look at aggregate stability. And one interesting thing is that even in the deeper soil, so this often is only done in the top 15 centimeters. So we see a huge difference in terms of the land that we left in the shrub willow and all the rest of the land that we tilled. Obviously, we've disturbed aggregate stability by doing that. But even our deeper soil, our 30 to 45 centimeter soil, had a strong impact. And that might have been the loss and decomposition of those roots underground. This was after 3 months. It was fairly interesting how profound that effect was, even with aggregate stability in deeper soils. And then soil pH, we got definitely very acidic soils, but the biochar kind of had a liming effect and a trend across the deeper soil. And with this being grass, the grass will root pretty deeply. So then we looked at shrub willow across a soil gradient. And for this one, we saw after 12 years of planting shrub willow, we had increases. So we looked at differences in initial soil organic matter. So very low soil organic matter, medium soil organic matter, high. After 12 years of planting shrub willow, we had over 50% increase of organic matter in our very poor soil, and around 25% in our medium to high organic matter soils. So shrub willow is a good way to increase soil health pretty quickly. This was just the surface soil though, because 12 years ago, they only took surface soil samples. So unfortunately, we can only compare the surface soil. But it would be interesting to know what was happening deeper. We did sample deeper this second time around, but don't have a baseline from the first time around. We also were looking at spatial variability. So our rows and alleys, so shrub willow is planted in a double row and then an alley. So every 3 years, they drive down to harvest. So you can imagine you might get some compaction, you get weeds in those alleys. So we wanted to look at spatial variation. We didn't see too much in the row and alley after this 12 years. There's a little bit of a trend in bulk density to actually see some compaction in the alleys. But this is another issue with these more complex systems, is this having to do the rows and alleys doubled our sampling, right? So we were able to sample a lot less because we had to sample in the rows and alleys. And this is something in these complex systems we often miss. We miss that spatial variability. Even in the agroforestry system that I introduced earlier, we sampled direct in the woodland, direct in the cropland, direct in the grassland, but we didn't scale that in terms of, well, in that system, how much cropland was there? How much woodland was there? Right, so if you don't properly account for that spatial variability, it's going to be difficult or impossible to say how much carbon you actually have on that land or how much really you're increasing soil health. All right, I'm going to skip this one, but if you're interested in soil organic carbon sources and like transplanting soil, which is what I called it, we put in C4 soil into the ground and are looking at microorganisms colonization of that. And again, looking at heterotrophic and respiration. Forest management. So we just started this, collected about 600 samples this summer. Had a great team, had a great time. Very challenging soils. So had to work on a lot of different iterations to get bulk density. developed what I'm calling mini quant pit. So this is a quantitative excavation pit, but is really the only way with these rocky soils to get bulk density. And it's quicker though than a full scale excavation. So trying to, again, balance this, being able to sample a lot. So they took a lot of samples, but do that efficiently, because we need to capture that variability, but also get critical measurements like bulk density. So we'll see how that goes. Looking pretty good for the measurements we got. I wanted to get, so generally into silvopasture, I'm not going to go through our whole project. We have a big initiative right now. A lot of folks from Cornell working on this. I'd love to talk about that. Basically, I'm applying all of the lessons learned from what I've just shown to this new silvopasture project, because you have this spatial variability. We want to dig deeper into these soils. We have a lot of rocks though. Methods in the lab that we need to deal with. And we're really looking at this system as a whole. So we really want to, understand, well, what does the producer need and want to know about soil health? And how do we get those results for them efficiently? And really speak to how that's helping the entire system as a whole. So we have a lot of different measurements that we're taking, including soil health, and trying to figure out what exact soil health measurements, how deep we want to go, how we want to measure bulk density, how many samples we can take and where. All of that is kind of being applied in this very complex system now. And if we can really expand our soil health framework to these other systems, then we can better show the evidence for how these more complex systems are improving soil health, and how much carbon, if there are carbon, strong carbon markets developing as incentives for farmers, if we're able to have them, calculate their change in carbon over time. So complex systems, I love this figure, very complex. So I'm just going to jump through. Shade is very important for lots of slides, but I want to get to questions. So this is kind of my last slide on thinking about all of that for these silvopasture systems within this framework. And finally, kind of just ending with that, looking to dig deeper, both literally and figuratively, into soil health, expanding soil health across diverse ecosystems. And really the ways that I see to do that are bridging basic and applied research, fostering these transdisciplinary collaborations to really understand that full systems view, and just learning, adapting, and embracing new ideas. It's more difficult as you kind of want to stick in your lane, but as you can learn more and be able to adapt, like especially I think everything breaking in the field on me all the time has helped with that ability to adapt to these new things, adapt to the opportunities, adapt to what those that we're working with, our stakeholders, need and want from us, as well as being able to answer those foundational basic science questions. All right, thank you so much. So thanks again for inviting me, and I'm so happy to take your questions and discuss further. Yes, hi, good to see you.
For your visit, really enjoyed your talk. I really enjoyed that you hammer in the equivalent soil mass concept, because I don't think it has really taken on as much as it needs to be, and probably still people take the same depth and just multiply bulk density with carbon contents, and that shouldn't happen anymore. I was wondering there's so many things in there that one can talk about. I enjoy that you also said that MEOM, there's a problem interpreting a MEOM fraction with persistence and durability, and I think that's exactly right. I think you can easily hang that very high and say, and tear down the MEOM concept. I think we need to rethink that concept. So MEOM becomes the new humic substance.
That's exactly what I've been saying. Yeah.
And that's problematic. But I was wondering about the diversity aspects and whether what constitutes for you diversity, and thus expanding the diversity of complex systems from just the soil to the ecosystem and the cropping systems. I mean, obviously, if there's an animal in there, it must be more complex and more diverse. But do you have a handle on what could you put on the x-axis when you have from low diversity to high diversity? Is it the photo where you see there's different colors in there or different heights of objects? What is diversity in an agro-ecosystem or a silvipastoral ecosystem? Or how could you measure that?
Yeah. Okay, so I'm going to remember to repeat questions. So the question is, I'm going to grab the end though. Yeah. So how do you measure the diversity within an ecosystem? What does that mean in terms of lower diversity to greater diversity and how that applies to soil health? Mm-hmm. Yep. I guess I do see it as when you do have more dynamic. So if you just kind of think about monoculture crop, right? It's all the same depth of roots. You have the same treatment year after year. You're maybe not applying anything to that. You're leaving it fallow. So just taking kind of the most commercial monoculture tilled each year. And I guess the diversity that I see comes in when, well, now you've incorporated animals, you've incorporated trees, so you have deeper roots. Maybe you've incorporated shrubs as well, so they're really filling in those different niches in the soil. And then you have poop in certain areas, maybe at high densities, lower densities. They do bale bombs as well sometimes, where you have, and I guess that's where that spatial variation comes in as well, as to how can you possibly sample all of that to get an idea of the entire system and what's going on when you have high carbon in one place, high organic matter, high soil health in one place, and then maybe less in another. So I guess I see the diversity that way of how many components you have within that system, the various differences in management, differences in inputs coming in year by year, even month by month. And then how do we actually measure that within the soil health framework? One of the big things is, I think that the soil health framework's really just based on the top 15 centimeters. And I think that at least to the 30 centimeters, if not deeper, is going to give us more information about how a soil and the soil functions are impacting the productivity of that land when you have roots that are extracting nutrients from deeper, tapping into deeper water. I don't know if that.
Yeah, and condensing that into one number. Right?
Yeah, I don't, yeah.
Yeah, to get that, measure it. And that's not easy.
Yeah.
Conceptually.
Yeah, well, I know even within the current soil health framework, condensing things into a single number tends to be a problem. And clouds, what could be only the couple things you need to change, but maybe overall it looks okay, but you actually have some real deficiencies that you could address.
Thank you.
Yep. Yes.
I was curious with the deep soil framework, like how operative it is in agricultural systems where you have a till layer and maybe the till layer is even impermeable. It kind of blocks water flow. So whatever DOC or DOM you'd want to see going deeper into the profile and shaking things up, that's not happening. So do you think, like, is it like the 15 to 20 or the 30 centimeter kind of conventional approach? Is that okay for agriculture? Or do you think even there we should be putting in the extra effort and break through the till layer and sample deeper?
Yeah, I think it's really dependent. So exactly, sorry. The question was in certain systems like agriculture, especially if you have a tilled layer that's really not letting water even filter down, roots can't get through it. Do we really need a sample deeper? Can we just sample to that depth? I guess my argument would be that we should be at least sampling to 30 centimeters in all systems. And then sampling deeper than that, I think, would be system dependent. I think that we should have essentially some sort of cutoff and framework as to what we should sample in all systems. And I think 15 is too shallow for agriculture. I think at least 30. I'd like to see 60 in agriculture too, but I think it is dependent on, well, a lot of our soils only go to 45. But I think it is dependent too on features, as you suggested. If there's real evidence that the deeper soil is not going to be affected, or that's not part of your question and this isn't going to go into a soil health framework, or is, but just isn't part of your question and you want to spend your effort elsewhere, I think that it doesn't have to be a golden standard to go deeper than 30. I do kind of hit firm at the 30 though.
Well, folks, we are out of time. Let's thank Cole one more time.
Thank you very much.
This has been a production of Cornell University, on the web at cornell.edu.